Hello students, in this lecture video, I am going to discuss about the important concepts of chapter 2, that is sexual reproduction in flowering plants. So here firstly let us see the flower structure over here. Let me zoom this. So here we know that a flowering plant looks like this. It has the colored portion which is referred to as the petal, and then the green leafy part is called as sepal. Then we have this ovary, and this is the stigma we can see over here, and a style. So this is the part of the female reproductive organ called as pistil. Then the male reproductive organ is the stamen that contains anthers, which inside those anthers there are yellow colored pollen grains and a filament is present. So together this is called as stamen. Collection of pistils can also be called as carpel. So then let us look over here that what is the significance of seed dispersal and food formation. It helps in colonizing the different areas, and many fruits they have mechanisms for the dispersal of seeds. Now there are actually a variety of pollination methods. What is pollination? When the pollen is being transferred on the surface of stigma, and then the pollen tube is being released from there, and the male gamete unites with the female gamete. So after pollination, these steps occur. Let us see what are the different types of pollination. It can be autogamy, ketonogamy, and xenogamy. Auto means self, where there is in the same flower self-pollination is happening. Getonogamy means that it is in the same plant. Okay, the flowers can be different on the same plant. Xenogamy means totally different. The pollen transfers from one to the different plant altogether. Then let us see the different agencies which are taking part in this, that is abiotic and biotic. Abiotic are the non-living components like wind and water. If it is wind, called as anemophily like in grasses. If it is water, it is hydrophily like Vallisneria and Hydrilla. In living organisms, it can be insects or birds. Insects, entomophily like yucca. Birds or gneithophily like daylilies. Now, here we can see that there are a variety of pre-fertilization events, means before fertilization, what is happening? Let us see over here. In the male gametophyte and female gametophyte, different male and female gametes will be formed. That is called as microsporogenesis, and in females, it is called as megasporogenesis. So in males, a microsporangium is formed, which you can see these four parts. In detail, here you can see epidermis, endothelium, middle layer, tapetum, and here this is the section of pollen grain which is being shown. Then in female gametophyte, we can see that there are, you can say, this ovary part is being shown over here, where the egg cell apparatus is going to be formed. Then, when we talk about ovaries, there are two types of ovaries, syncarpus and apocarpus. Syncarpus, where it is fused, and apocarpus, which is free. Then we can see the different devices which promotes the cross-pollination, that is self-incompatibility, which will prevent the self-pollination. Arrangement of anther and stigma at different positions so that they cannot do self-pollination. Then in some plants, it can be the production of unisexual flowers only. Then it can be that the two processes, that is pollen release and stigma receptivity, these are not synchronized, means they occur at different time periods. Then let us understand about pollen-pistil interaction. So in this, basically the pollen release and stigma receptivity has to be synchronized with each other. Let us talk about some special modes of sexual reproduction. It can be parthenocarpy, which is development of fruit without fertilization. Polyembryony, where more than one embryo is produced. And apomixis is production of seed without any fertilization. Now let us see what is happening in fertilization. There is double fertilization events where there is syngamy, that is the fusion of male and female gamete, plus the fusion of the central cell to form the endosperm. So it is called as double fertilization. Then there can be two types of seeds, albuminous and non-albuminous. You can see over here two different types of embryos also, dicot embryo and monocot embryo. Their structures are totally different. Then here coming to the different types of fruits which are being formed, it can be true fruit and false fruit. True fruit, it develops from ovary, whereas false fruit, it develops from any other part except the ovary. Then, in case of endosperm, we can see that sometimes it provides nutrition to embryo as well as seed also. Okay, then it is albuminous. And if it is not present in the seed, then it is non-albuminous. Lastly, let us see that how artificial hybridization is happening. It is one of the approaches where we need the crop improvement processes. It can be achieved by emasculation, that is removal of anthers, so that we can decide which pollen will be released on which stigma. And bagging is that we cover that anther, we cover the stigma basically, so that we know that when that artificial hybridization process has to occur. So this is just a summary of the chapter